What is a genetic fact that not a lot of people know? Story 1. There is a gene called TP53. It's referred to as the guardian of the genome. It is a tumor suppressor gene and prevents cancer of all types. Humans have two copies, and if one is broken, mutated, it causes an inherited condition called Li-Fraumeni. People with this have a very, very high risk for cancer, and the condition can be passed down in families. Elephants have 20 copies of TP53 and therefore rarely get cancer. The Li-Fraumeni Foundation uses the elephant as its mascot, and hopefully we can someday figure out how to replace faulty TP53 genes for these families. Actually, what's also interesting is that having a mutated copy of P53 leads to worse cancer outcomes than losing it altogether. Therefore, although it's thought to be a tumor suppressor gene such that losing it leads to cancer, it might also be considered oncogenic. Story 2. Since navel oranges have no seeds, they are all clones of the original mutated fruit discovered by a Brazilian monk a century ago. The billions and billions of navel oranges that have been grown, sold, and consumed since are all genetically identical. So cool, there's a farmer in Australia that somewhat recently discovered a naturally seedless lemon tree in his grove and is now making money off of the clones, I'm sure. Red flesh grapefruits are the product of using radiation to trigger mutations and see what happened. There was a little trend called atomic gardening where they'd even sell kits of cobalt-60 for people to try to create cool mutations on their own. Story 3. If you set aside ethics and set out to do it, it would only take 33 generations to create a human being who was the descendant of everyone currently alive and able to have children. Also, pedigree collapse. No living person has as many unique ancestors as they mathematically should. Every person has, biologically, 2 parents, 4 grandparents, 8 great-grandparents, etc., doubling every generation. 30 generations back, without any degree of inbreeding, would require over a billion unique 28x great-grandparents, more than the entire world population for 1100 AD. Every single human's family tree is full of people marrying and having children with cousins of some genetically unimportant degree over the last 1,000 years. Story 4. Genetic chimera is arguably the most interesting condition. It is possible and documented for a woman to give natural birth, but the baby does not match her DNA test. Essentially, 2 fertilized eggs merge at a very early stage and develop into a single organism. Different parts of the body consisted of one or the other genome. It's most commonly discovered in domestic animals that have unexplainable color combinations, often but not always, divided symmetrically. Story 5. When you're pregnant, bits of DNA from your baby invade your body and settle all over permanently, including in your brain. Researchers found this by looking for male DNA in the brains of deceased 70-year-olds. Interestingly, mouse studies have also shown that fetal cells within the mother have restorative properties and will rush to places like the heart, kidneys, lungs, liver if it's injured, and can even become cells that begin beating. One of the theories I have seen, but I don't know if anyone knows for certain if this is true, is that these cells enter our bodies, so a host is less likely to reject the parasite baby. Story 6. Most genetic studies were, and are still, done on white Western populations. Some things that we know about genetics turn out to not work so well in non-white or non-Western populations. Loose example, diagnostic tests to check for certain diseases sometimes misdiagnose Americans with African ancestry because sometimes we don't actually know exactly how certain diseases will look in non-European ancestry patients. We just assume it will be the same. Africa as a whole is the most genetically diverse continent for human genetics. Humans outside of Africa went through a population bottleneck, resulting in an overall lack of diversity moving forward. The human blender of genetics in Europe just didn't have many starting ingredients. There are things called linkage disequilibrium maps, which basically map out sections of the genome that are usually found inherited together. In Europe, these maps are quite chunky, with large identifiable sections that are sort of reliably found intact. But in Africa, the blender was always running and never had a bottleneck, so those maps are basically a much finer puree with a lot more mixing. This presents certain challenges, as many studies rely on those maps to make predictions, and it's harder to do that when the map for Africa as a whole is basically way more complex. Everyone's cancer is its own unique genetic disease. There is no one true single disease called lung cancer, just as there is no one single true human genome. Everyone is genetically unique, yes, even identical twins, and every cancer is unique. Story 7: 1 in 1,000 babies will be born with balanced chromosomal translocation. Women who carry it, or partners of men carrying it, will typically have more frequent miscarriages. My MIL had 5 or 6 miscarriages because my file carries balanced chromosomal translocation. 2 of their kids inherited it, the third has normal chromosomes. Balanced chromosomal translocation means that 2 chromosomes switched a part of their chromosome, but it was an even exchange. For the people who inherit it, it means they could get pregnant or impregnate their partner with a baby with unbalanced chromosomal translocation. Those babies usually miscarry. Story 8. UV gives you cancer because literally makes your DNA stick so tightly the only way to fix it is remove the entire section, then guess what was there. More this happens, the more chance a cell becomes cancerous. Bacteria don't have this polymerase repair, so it's why UV kills them or mutates them. Plant 1. Hexaploid wheat possesses 42 chromosomes derived from its 3 ancestral genomes. The 21 pairs of chromosomes can be further divided into 7 groups of 6 chromosomes, 1 chromosome pair being derived from each of the 3 ancestral genomes. This is mainly because of how much humans have bred it. It's literally inbred. Story 9. Perhaps my favorite biology fact is about pharmacogenetics, how your genetics determines the efficacy of drugs on your system due to prevalence of receptors of different types that the drugs target to produce an effect. 90% of drugs only work on 30 to 50% of the population. I find it so truly wild, but most people I've talked to about it seem to agree that some drug or other doesn't work for them. This really highlights how much of a process finding the right drug for a person is. I had a lecture on this in uni in about 2016, and it said the future of pharmacology is individual genetic screens for drug effectiveness, meaning people don't have to keep trying different drugs until they find the one that works. You would be tested, then your doctor would have that info. I just hope it overcomes the pushback from Big Pharma in my lifetime, because it would be a true game changer. Story 10. White tigers are not a separate subspecies of tiger. They are just super inbred. About 1 in every 3 white tigers also have some pretty horrible health problems that come from the inbreeding process, such as cleft palates and crossed eyes. Not to mention the people that are purposefully inbreeding tigers in order to produce a white tiger end up with quite a few normal tigers that they don't necessarily care for or want. It was a really big problem years ago, especially in not super reputable faculties. Roughly 10 or so years ago, the AZA, Association of Zoos and Aquariums, made a statement about not allowing the breeding of animals specifically for coat variations, so all white tigers, white lions, king cheetahs, etc. that you see in an AZA-accredited zoo were not bred there and came from a different facility. White tigers do occur naturally, super duper rare, but are often killed by mama tigers when they are still very young. The white coat sticks out like a sore thumb in the jungle, and she kills the white cub to protect her other cubs from danger. White lions are in the same boat, but there is a population of them that occur naturally in South Africa. The ones you see in non-reputable exotic animal facilities, roadside zoos, are all inbred for the purpose of that white coat. Story 11. Pericentric and paracentric inversions. When meiosis happens in our cells, something called crossing over occurs, which is where the chromosomes line up, break apart, and recombine. There are tons of ways this can go wrong and is how we get genetic deletions, duplications, and translocations. Inversions are kinda neat because a segment inside of a chromosome ends up flipped upside down. It's usually not noticeable since all the genetic material is present, but when meiosis happens, things really go bad. It turns out chromosomes know how to pair up by markers all along it, and if a piece is upside down, then one of the chromosomes has to make a loop to line up properly. If crossing over happens in the loop, then you get chromosomes that are either totally messed up, para, or deletions, duplications, peri. This causes infertility since you have a reduced number of viable cells, and it's entirely by chance, not a set percentage of normal versus abnormal cells like a regular mutation brings. Story 12. Trauma can be inherited. Look up transgenerational trauma. So if you feel more sensitive than most people, it might be because your grandma experienced something horrible. On average, it takes 3 generations to heal a trauma, if I remember correctly, but only if properly treated, and the parents do not re-traumatize their kids with their unconscious trauma responses. This way, unhealthy behaviors would become the norm and eventually become genetically preferred automatic behavior for future generations. Story 13. While you inherit 50% of your genes from each of your 2 parents, that does not mean that you inherit 25% of each of your 4 grandparents' genes. Genetic randomness results in inequities in second-generation percentages. So while you have 50% of mom's genes, whose genes are, in turn, 50% of each of her parents, that 50% might actually break down to 40% of maternal grandmother genes and 10% of maternal grandfather genes. In cases where the genetic distribution is more equitable, the grandchild might look like a mix of several grandparents, or he/she might not look like any of them. The result is that in the second generation, you might have a progeny that strongly favors a single grandparent. Genetics are absolutely wild that way. Story 14. Mosaicism is when someone has genetically different cells across their body. For example, someone could have a trisomy for the 21st chromosome, Down syndrome, in some cells, but not others. There is also something called confined placental mosaicism, when the cells of the placenta have the genetic condition, but the fetus does not. Sometimes it happens after the cells become the fetus and the placenta have already separated, and sometimes before they do, which is usually when you do see the genetic condition in the fetus. But there is also something called trisomy rescue, where the trisomic cells may die off, and the diploid ones continue. Story 15. The coat color of cats is X chromosome linked. All cells should only have one active X chromosome. In females, one X is randomly deactivated early in embryonic development, so females end up with clumps of cells expressing one X, another clump of cells expressing another X. This is why most calico cats are female. The different coloring regions are where different X chromosomes are activated. If you have a male calico cat, they most likely have polyploidy, or XXY chromosomes, but they are pretty rare. Also, a chromosome mutation can affect the expression rates of genes on completely different areas of the chromosome. This happens because a mutation can alter the folded structure of the chromosome. In some cases, the more exposed a gene is to the transcription machinery of the cell, the higher its transcription rate. Something happens that causes the chromosome to fold differently, and now that gene is in the interior of the fold, less transcription, and vice versa. Story 16. CCR5 delta 32 mutation, hypothesized to have conferred immunity to the plague during the Middle Ages, so conserved in Northern Europe, now confers immunity to HIV infection. A man was cured of HIV when he was given a bone marrow transplant to cure his leukemia with bone marrow from a person who was homozygous for the delta 32 mutation. Unfortunately died recently, if I remember right, due to cancer recurrence. Story 17. A little more than 100 years ago, we had a strong scientific consensus on the efficacy of this genetic science. Its supporters included Theodore Roosevelt, Woodrow Wilson, and Winston Churchill. It was approved by Supreme Court Justices Oliver Wendell Holmes and Louis Brandeis, who ruled in its favor. The famous names who supported it included Alexander Graham Bell, inventor of the telephone, activist Margaret Sanger, botanist Luther Burbank, Leland Stanford, founder of Stanford University, the novelist H.G. Wells, the playwright George Bernard Shaw, and hundreds of others. Nobel Prize winners gave support. Research was backed by the Carnegie and Rockefeller Foundations. The Cold Springs Harbor Institute was built to carry out this research, but important work was also done at Harvard, Yale, Princeton, Stanford, and Johns Hopkins. Legislation to address the crisis was passed in states from New York to California. These efforts had the support of the National Academy of Sciences, the American Medical Association, and the National Research Council. It was said that if Jesus were alive, he would have supported this effort. All in all, the research, legislation, and molding of public opinion surrounding the theory went on for almost half a century. Those who opposed the theory were shouted down and called reactionary, blind to reality, or just plain ignorant. That science was eugenics. Many nations, including the US, sterilized what the science determined were less than genetically desirable humans. The Germans used this genetic science to justify their concentration camps and mass slaughter of people. That bad science is called eugenics. Story 18. Red hair is usually caused by a mutation in a specific signaling pathway, where a protein called ACIP binds a receptor called MC1R. MC1R is on melanocytes, which are pigment cells, so changing this pathway changes how pigmentation is regulated, so linked to red hair, freckles, etc. Because of the change in pigmentation, redheads tend to be lighter skinned and more likely to have certain conditions like light hypersensitivity. But here's the crazy part. Lots of other cell types besides melanocytes have MC1R receptors as well. Consequently, red hair mutations are linked to changes in bleeding and pain sensitivity. The details of how this works are active areas of research, but this seems to be a broadly conserved trend in many animals, so it seems likely to be a general trend, meaning red-haired individuals in other mammals might bleed more and feel more pain. So there are clear examples of where hair color makes very specific predictions about seemingly unrelated aspects of health. This is not a purely theoretical matter, but is a practical consideration during surgery. Story 19. As you could probably deduce, tumor cells from cancer have their own mutated DNA that differs from your real DNA at some crucial location, leading to out-of-control cell division. When these tumor cells die, their DNA is sloughed off into the bloodstream. DNA sequencing technology and computational power have come so far that we can find single digits of mutated DNA bases from the tumor if we take a patient blood sample. To simplify how crazy this is, consider that one copy of your whole human genome is about 3.25 nanograms. In 5 millimol of blood drawn in a tube at your doctor, there is about 25,000 nanograms of your DNA. That comes out to 7,700 copies of your entire human code. In those 7,700 copies, there are 23.1 trillion A, T, C, and Gs, and we are at the point where we can find the single digits of bases that don't belong. Story 20. Genetics make up the bulk of why any person is capable of going professional in any given sport or skill-based thing. You talk to most people and they think it's hard work or anyone can do it, but the fact is, in many things, millions are working hard and trying to get there, but only those with the right genetic gifts will make it. The main issue is that most people also don't realize it's not just one genetic trait that makes you that good. You need to stack many times. For instance, most basketball players are taller than average, but they also have other things like stamina and coordination that are less well defined. There are many very tall big players, but only a few have all of what it takes to join the NBA, and many tall people have fallen out because of lack of coordination or strength or other issues. Most people use that as an explanation of why its genetics are completely wrong. Even your mental side of things is also genetic. Some people are simply more motivated and driven, while other people simply aren't. Story 21. It's more complicated than they teach you in school. Genes can interact in ways that you wouldn't predict. Two blue-eyed people can have a brown-eyed child, and it doesn't absolutely mean that cheating was happening. Basically, genes can be suppressed by other genes, or by their absence. Environmental factors can activate or suppress genes. Gregor Mendel, who basically pioneered the study of genetics, actually cherry-picked his results because of some outlying data that didn't fit the model. They tried to breed calm foxes once to make farming them for their fur easier, but when they achieved calm foxes, the foxes developed floppy ears and spotted coats. Turned out that the reduced adrenaline caused other genes to be activated or not activated or something like that. Edit. I looked it up. The experiment is still going on, and the results have been very successful. They're at 58 generations now. With androgen insensitivity syndrome, you can have a Y chromosome and produce testosterone normally, but an error elsewhere in your DNA causes your body to be immune to testosterone, and so you develop as female. In short, there are many cases where genes work in tandem, and an error in one place can have unexpected side effects. There are many cases where a gene doesn't do anything unless the environment is right for it. During development, certain genes need to be triggered at different times and places for the fetus to develop. The most well-known example of this failing was the thalidomide disaster, where the drug suppressed certain genes at the wrong time, resulting in limbs not forming during gestation. There's a mutation in fruit flies called antennapedia, where the leg gene gets activated when the antenna gene should have. Story 22. If you have red hair, you likely have a naturally high resistance to anesthetics and other depressants. Surprisingly, not a lot of research has been done into this link, but they think it may have to do with the MC1R gene, which is involved in melanin expression. I'm not a doctor. While some studies have been done, the knowledge is mostly anecdotal evidence. Ask an anesthesiologist, though, and they'll probably tell you they usually need to increase the dose for ginger patients. Interestingly, I have this gene even though I have dark brown hair and Mediterranean skin. I was born with red hair, but nobody in my family has been a full ginger for at least a few generations. I needed teeth pulled as a kid, and the dentist needed something like 10 shots of local anesthetic to numb the pain. I had surgery and counted down into the 80s without falling asleep and kept moving my head to let them know I was awake. I did ketamine in college, and it had zero effect on me until I did a half gram all at once, which I don't recommend. It gets frustrating when I need a prescription for Ativan or when I've needed painkillers, because when I tell them I need a higher than average dose for it to be effective, they usually think I'm trying to get high. This is a tangent, but I also have ADHD, meaning stimulants don't affect me normally either, and usually have the opposite effect. So except for psychedelics, I don't experience most drugs, like normal people do. I'm genuinely curious what the odds are of carrying that redhead gene for depressant resistance without it being expressed visually and having ADHD. Story 23. There is a gene that determines how you will react to narcotics. It should be tested for free before the patient is prescribed opioids, because the doctor will be able to see how you will react to it. Group 1 gets the normal amount of pain relief, and it's very rare for anyone in it to be addicted to opioids. Group 2 receive a so-called wild effect from even the smallest of dose, meaning they get high from it and will become addicted almost immediately, leading to harder drugs, and this group should never be prescribed narcotics. Group 3 receive no pain relief whatsoever from opioids, no matter how high the dose, which means that they genetically will not become addicted because it's useless to us. Yes, I'm in this group, which sucks. CYP2D6 defect is what needs to be tested. Story 24. About 8% of our DNA is not human and was inserted there by ancient viruses. Another 40% is made up of DNA that acts like a virus but can't leave the cell. These are called transposons, and they can copy-paste themselves. These transposons are thought to be of viral origin. They inserted themselves into our DNA in order to do what a virus does and replicate, but lacked the machinery to get out, so they got stuck. Story 25. Tissue wounding, even when it heals properly, i.e., no scarring, fibrosis, can leave an epigenetic memory in the cells of the tissue. The stretches of DNA that encode genes that help the cells respond to and heal the wound remain open and ready to respond to future wounds, even though at the level of RNA transcription, the unwounded and properly healed tissue are largely indistinguishable. This helps the tissue respond rapidly to future wounds, whereas during the first wound, a cascade of molecular events leads to the DNA unwinding opening of these wound response genes. Story 26. Lots of research rooted in justifying scientific racism works backwards. A 1992 study by Columbia University's Lowenstein Center for the Study and Prevention of Childhood Disruptive Behavior Disorders wanted to identify genetic markers for violence in black children. To do so, they ran a study on the younger siblings of incarcerated teens. Their experiment on the children produced results counter to their initial hypothesis, so they changed the hypothesis and published the study as evidence supporting the idea that violent behavior was a genetic predisposition. And why not? All the children were related to delinquents. That they were poor or living in single-parent households didn't factor into the study. Story 27. You are part virus. There are generic regions, elements, human endogenous retroviruses that are remnants of old viruses that infected our population, like HIV. Eventually, the genes of the virus degraded, but they're still there. Similarly, our mitochondria have their own genome. They were bacteria, captured by the earliest eukaryotes and converted into the energy factories of the cell. They have different versions of some of the same genes in our nuclear DNA.